Nice to visit all the friends again. I work with biological physics also, and in fact, I have it's a collaboration between the Institute of Physics of Rio Grande do Sul, but I also work at Universidade Federal of Rio Grande do Norte, which is here, but it is in the bioinformatic postgrad. Okay, the idea is we started to do this. Well, this is a collaboration with some students and some people that are working in biological physics, and then some collaborators in Indiana University and Paris Diderot, and also some colleagues at the university and people from the bio institutes in Rio Grande do Sul. So, first, I have to talk why we do. Although many people argue with that, but I'm still a physicist, although we are studying biology. And the point is that you have nature and mathematics, and that's what breeds science. I know it's a little bit controversial, this thing, but anyway, that's the way I do it. And the idea, you've got a nature, then you have phenomena, you don't have numbers, and you have a dynamics, and eventually your system will go from state A to state B. And what you do is that you describe your phenomena with measurements to transform these things in numbers so you can do mathematics out of it. Okay, and then you have a model. And if you have a good model, then you can do something which is the modeling of your system. Of course, your modeling is never perfect, right? It depends on what you want. And what I say is that when you do science like this, you are doing physics. It doesn't matter if the phenomena are biological or sociological or really solid state or something like that. Okay. But when a model is a good one, when a model is an interesting one, okay, when it answers your questions, so you have always to make your questions before saying your model is good. And the idea you should make good questions. If the model does not discriminate between competing explanations for the phenomena you are focusing on, this is not a good question, so you won't build up a good model. And you have to collect from the model more than what you have planted, right? Because then you really have profit of it. And sometimes you have to use the Occam's razor and keep it as simple as you can. Although in biology, sometimes this is not always completely as true. And you have to understand what are the necessary ingredients but are not enough, and which ones that are enough hypotheses, but they are not unique. In biology, this is very common. I used to say to my students that when you have one explanation for a biological phenomenon, and you have another explanation, probably all the 5 explanations you can think of are true. I mean, biology really finds its ways in being redundant. Okay, so you have to understand that to be careful and make models of biological stuff. We also have something which is quantitative and qualitative, okay? When you're doing a model, it's very common when you are doing biology or models for biology that you are very happy in doing qualitative models, and this is very good, but it's not enough. Not if you want really to make some difference and solve some real problem in biology or in therapeutics or just helping to find, I don't know, something which is very important, okay? So qualitative is important, and the first thing that you should check, do you have an equilibrium? Do you have stationary states? Are they self-similar? Which kinds of stability you are observing? Chaos, attractors. But qualitative allows manipulation. If you don't have quantitative things, quantitative predictions, you cannot really engineer some solution, okay? So how to set a system to behave the way you want? Not your system, not your model, but the real system. How can you beget therapy for that? How to gather information from your observations? Well, the point is the measures, and this is how we can contribute when you are a physicist. Measures that is necessary for you to make your system, to make your model, they most frequently depend on the model you are assuming, okay? So unless, I don't know, about, just counting the number of a given length, you can put in the length of this table. This is a very, very simple thing. Still, this is a model. But if you have any other thing, if they are not the fundamental quantities that you use to define the basic units of your measurement system, then you must have a model for that. And now this is the point that I want to apply what we are doing. And I'm going to explain you some things that we have applied for cell migration. The idea, the whole thing started when we tried to measure and understand how a collective of cells may migrate. And in this way, we started to see how people were measuring and how people were comparing and how people were simulating. And we came across with this thing that they were not properly defined. The measurements were not well defined in a way that you could use quantitative results to predict what was happening. And if you don't do that, you cannot characterize correctly. So what I'm going to present here is the first part, the results of a first part of a project that we are doing, that our aim is to describe collective cells migrating in a way that I could eventually write the biological fluid equations, okay? Which is something, but it's a holy grail of the field, but it's something that you could do trying to write some Navier-Stokes version of what's happening when you have a collection of cells migrating, which is very relevant if you want to understand wounding phenomena or development phenomena. I mean, when embryo is forming, or when you want to understand how to prevent some metastasis of or cancer development. But I need numbers for that. So we decided to do it very, very careful. What I can tell you is that, well, usually the observation of cells migrating under the microscope over a flat surface is very old. I mean, it's about or more 100 years old, okay? And the first thing that people realized is that they were a persistent random walk, okay? A persistent Brownian motion. So what you have is a Langevin equation. And when you have a Langevin equation, you have the velocity of the cell, the VDT, and a friction term, which depends on V, and then a white noise there. And then you solve that, okay? It's a white noise, zero mean, and non-correlated. Okay, so I can do this. Okay, oops. And then you have the Orsten-Uhlenbeck process, which is, in fact, the Langevin equation. And when you consider the one dimension for simplicity, suppose a cell at the origin, and then you take the dissipation, and then you have this equation, you solve it, you have the green function of it and everything, and then a white noise is there. And then you can see what's happening. And then you have all this equation here, which is the velocity for one given realization of a sequence of possible noise terms. And then you apply the average over all the noise. And then what you have is something which is like that, which will give you, because you are interested in this. And then you have the velocity, and this is a very well-known result that even, Einstein has published an equivalent equation when he was doing that. And the idea is that here you have the assumption that your particle has already equilibrated with the bumps that it receives from the noise, and then this thing is zero, and then you have something which does not depend on time, because this will be zero, and then the average will be just like that. And assuming that this is the average kinetic energy and its temperature, you can have the density, the velocity, okay? Well, you can also do something which is the mean square displacement, which has been explained so nicely before. You can just calculate where the cell is and where it will be if you wait delta t. And then you calculate all the same thing. And then you arrive with this equation here, which is the fourth equation, what you call, because the fourth was the biologist that was looking at this. And you see that you've got something that first you can rescale it with the natural units. P is the persistent time, in the sense that when the time you wait is much larger than P, this thing disappears. And what you have is something which is the mean square displacement, just go linearly with T, which is diffusion, okay? And if you have something when delta T is very small, you can show that this thing goes with delta T as delta T to square. So you have a ballistic thing, and then you have a transition to a diffusion equation that's whatever everybody was expecting to do. And you can write these equations. You can write these equations in without telling any, without any parameter, you see. When you use all the parameters, you put in the equation. Here, you can use this parameter to write the natural unit of length and the natural unit of time, and then you have a universal equation. Oh, that's wonderful. That's what you're going to use to, if it's true that this is the first equation. It was 1919, okay? And it has this appearance, which is a ballistic behavior, and then a change for a diffusive behavior. Okay? Okay. That would be very good. That would be very good. And the idea was just, okay, I take my cells, and I measure some trajectories, and I measure the mean square displacement, and I put it in the plot, log plot, and then I try to fit this equation over. I change all the things. I look for these equation, for these unities that will collapse my results over the theoretical curve, and then I have the values of the quantities, the parameters, the fusion equation, everything, and then I'm really measuring my thing. The point is, this is how you do the experiment. You do some trajectory, and then you will measuring all the things, so too early. And this is the results from from 4 different experiments that we just got from the literature. Okay? In the dashed line here, what you have there is the fourth equation. This is the ballistic behavior, and this is the non-ballistic behavior. I mean, this is the ballistic, this is diffusion, right? And these are different different experiments, and from cells just put in there, migrating as single cells, and they have been recorded for a long time. And that's what they got. And when you just try to collapse, what you see is that there is some initial deviation, which is not predicted by the fourth equation, okay? And also, I mean, this was very disturbing for us, because we were trying to build a model that I'm going to talk after that, a simulation model, to put all the cells migrating, see the collective behavior from these cells that would reproduce the single cell behavior, and then see what I must add in the system in order to make it behave just like what I'm observing in the collective. Well, what happened is that we could not understand really what was happening. And more than that, if I don't have these things, if I didn't have these things, there were many different ways that I could collapse these curves. So, and then you say, well, what people would say, well, they just considered it a measurement error. But it is not. I mean, you just go, and people were, some of the experimentalists were saying, no, they cannot be, but I don't know what it is, and I'm not going to pay attention. And the other says, well, maybe a segmentation error, because you are taking pixelated images, and then maybe it comes from there. No, it doesn't come. I mean, you can make all the calculations. And what happens? Well, in this fourth equation that we had there, we just added a parameter as we just added one parameter here that would tell you at what fraction of this time, which defines the transition between the ballistic behavior to the second diffusive behavior, and at what fraction of this persistence time that the system goes from a first diffusive behavior to ballistic and then diffusive again. And this is what's going to be different from all the cells that we could do. And when you do that, you can put all the systems, all the equations that you had here. You can look at all the equations here. You can fit, again, without any parameter here, because I use the fitting to define which is the length scale and which is the time scale. And the only difference between the different cells are at the fraction of the persistent time that it links being a first diffusive problem. Okay? Long times, short time intervals. I remember that this is not a long time, right? This is not a long time. If delta tau is long, it doesn't mean that it's long in the experiment, but it's long interval between where I'm measuring the first cell position and the second cell position. And then I do it again, and then I do it again, but it's already equilibrated, okay? Initial diffusive regime is a problem for velocity definition, and this is the problem. Because you see, the definition of velocity is the variation in position over variation of time. And if you go to very small time, you should even be, you should be more, each time, more precise about your velocity. But this is not what happens if you have the first diffusive environment with a different initial length for the first diffusive regime. Then for each experiment, choose a very small time interval. You do it wrong. Because if you do that, you are going to measure things that are indeed, they are diffusing and not in a ballistic regime, and there is no velocity defined in that regime. So, how to measure? What should we do to make a definition, to make a characterization of cell migration? Because now, if I have, yes. Yes, that's a very good question. But the point is, the experimentalists were classifying their cells as more rapid or less rapid, and they cannot do that. And they were trying to see if depending on the velocity they are assigning to each cell, if they could discover what was happening to all the mechanisms internal. So this you cannot do because it's not well defined. You have to have first a good definition of the measurement that you are going to do for your cell. And that means that you have first to have your model and then to define your measure. Okay? So what is, the second question is, okay, I understand where the fourth equation comes. It comes from the Langevin equation with the, but what is the equation that will give that modified fourth equation where we have added the ester? Okay? What we have done, and this is Mendele, Marcia, who has done, we still have to write it, but it's amazing. It's not I mean, what would you do? I mean, we started doing, I'm just going to consider another kind of source of noise that would be faster. But you cannot do it. If it's a white noise, they will just sum up, and they will be mixed, and you are not going to see anything. What you do have to do is a second uncoupled source of motion, source of noise. And what you do is that you have to have first, you are not going to see this thing if you're not going to, you have to consider a two-dimensional motion, which is in the plane, okay? And the idea is that the cell is polarized, and it is, because if it's not polarized, it cannot migrate. The cell doesn't have a, will to go do this and this, and I go there. It depends on how the actin cytoskeleton is organized there. So it has a preferential polarization that will make it go to there. But it's a very noisy situation, and then they have some, lateral source of noise. And what happens, what we could do, could come from the model, is that something, you have this velocity, which is the parallel to this polarization, and the noise that is parallel for this polarization. And this follows the fourth equation. But you have something which is a random orthogonal displacement that goes there, that comes from the noise of reorganizing. This is stochastic dynamics of the actin inside the cell. And what happens is that overdamped motion in that direction, so it goes like that. But after it has really moved, I mean, the movement is not only this one, but it is the displacement that this implies, summed with the displacement that is the random orthogonal noise. Then, when you do this, which was the real direction that the cell has moved, then you redefine which is the polarization axis. So after one time, the parallel velocity here, it's not calculated in the same axis as a time before. And when you do this, then you can obtain exactly the same curves. Yes, yes, it's also white noise. And I keep saying that this model means that the cell migrates as not as a drunkard, but as an obstinated drunkard. It looks at you, and then it goes there, and then it looks at him, and then it changes. Okay? But it's, and what's going to make me change is just the internal fast noise that you have just messing up with the definition of the actin orientation inside the cell. So what's important here is that we have defined the time. What we have shown is that the time at which the cell changes the orientation, which means that if I don't look to this very small time scale that I'm conserving, persisting in my direction, and then I change, during this time, I cannot consider all the time ballistic as it was considered before. I had this signalization that it had a fast scale thing that make you like that, but this fast scale probably is the cause for the change in direction. So the very, very fast at a small time is changing the behavior of the cell at long times. Okay. So then we have the parallel to theta, the perpendicular to theta. Theta is the direction of the real velocity. Okay. And then you have a term that tends to realign the theta, the orientation of the acting direction there, to the current velocity vector. And when you do this, you just obtain the same thing that you should have. So internal orientation is necessary to explain the observed short-time diffusive regime. You cannot have it if you don't add these new variables to the system. And what is S that we have put it there? S grasps the effect of the internal dynamics in producing movement fluctuations perpendicular to the previous movement direction. Persistent time and maximal exponent in the MSD versus tau depends on that. And cell polarity should have something to do with cell speed. I mean, these are, although you can say that up to now, all the definitions of cell speed are not really well done. Cell polarity was because of that. I mean, you look to a cell, okay, and you see they have all this lamellipodium, which makes cell want to go there. And then the biologist will tell you, no, listen, the direction of the polarization, it's not always defining where the cell is going. But the point is, if you don't know how to measure velocity, and you try to take even smaller and smaller delta t, you are going to find something that the polarization of the cell and the trim, you know, the wiggle that it does, it's not correlated. And so this is the problem. So what we have here now is that we need a polarity definition. We have a speed measurement protocol, and what are the correlations between them? This is important for my ultimate goal, which is the trying to model all this collective behavior. Because it's true that I can follow some cells and make the experiment and see what's happening to it, and looking how the cell is, you know, elongated, where the actin is and where the nucleus is. But I cannot do that for 1,000 cells in every second of my system. I mean, I have to have some a better way to measure where the cells are going when I have a very large number of cells migrating together. And sometimes they are doing some, vortexes. And if I do vortexes, I'd like to be able to measure the field of velocities. The idea is to correlate the polarization in shape of cells with the velocity of cells. And for doing that, I have to first understand the correlation between polarity of shape and the cell velocity. So, let's do it. Let's first do it in simulations. What we have done is just to simulated some three-dimensional cells migrating over a two-dimensional flat surface, which is the first thing that everybody does. So, I have first to know if the model that I'm building up is doing exactly the same as the experiments they are doing, okay? And what we have done is just to use the CompuCell 3D environment proposed by the group of James Glazer at the Indiana University at Bloomington. And the idea is that these are POTS cells. They are based on the cellular POTS model, which means that if I have a domain with the same POTS label, then I have one cell or one compartment of the cell, okay? And then I use as an energy function one thing more, which is a volume term that will guarantee that the cell can fluctuate volume, but not very much. You are just going to see. And what we see is that if you start with a very, almost spherical cell, then what happens is that it goes there. It's just the same thing. It's just that you are seeing a slanted image. And then these in yellow we are modeling a very rigid nucleus. And then you have the cytoplasm, what we call. But when the cytoplasm touches the substrate, it has some probability that will create something that can push the cell. But it's just, there is no, in the Hamiltonian or in the idea that we are doing, there is no way we are prescribing where the cell should go and what is the polarization. You have here something which is polarized. It starts rounded like that. It polarizes because all the lamellipodium goes in one side, and then the cell starts migrating to there. I'm going to explain how we managed to do that. And what we have considered is an interface energy. It's an interface energy which is just the usual POTS model energy. It depends on the cell and the compartment. The cell, because we are trying to make all the things for very large number of cells, but now I'm just showing you the simulations for a single cell, so don't pay attention in the S. But the C, it will be 1, 2, or 3, depending if it is nucleus, cytoplasm, or front, or lamellipodium that is responsible for pushing the cell. And then we have the target volume that says that the volume, the energy will increase if the volume of the number of pixels that you have in each compartment is different from that one that the cell knows by DNA or something to have. And then we have something which is new in this model, which is what is responsible for dragging the cell. This thing here we call an F-actin. In fact, it's an artifact that we do to make to simulate the fact that the cell has this probability of forming the lamellipodium, which is a mesh of actin filaments, and the filaments keep on growing very near the membrane, so they are always growing there, and they are disassembling in the rear. So although almost each one of the monomers are not moving themselves, the center of mass of each one of the polymers are moving forward because you are putting there and you are disassembling here. And sometimes they, of course, make some attachment to the substrate because that's the way they are not going to be pushed back. Well, to do this, to simulate this, what we consider a field, okay, the action diffusion equation here for the F-actin field. There are some sources in every single pixel of lamellipodium, which are the gray thing, and they decay very, very fast if they are outside the lamellipodium, which is also true because these filaments are not surviving outside the lamellipodium. And then what we say is just that when you choose two pixels to copy one over the other, this probability of copy is going to be enhanced if you have a gradient of the field like that. And then I do it, increase the probability of increasing here. But if it increases there, it increases its volume. And then the target volume term is going to decrease the probability that will keep there. So if the cytoplasm wants to grow over the lamellipodium, then it will grow. And the same happens here, because if the cytoplasm has grown here, then it will tend to decrease there when you have the same thing. So you have a polarization axis. And that's what we got. And then we have a film, right? Here. And these are periodic boundary conditions. So when you see something, this is the same cell. So it goes there. Sometimes the cytoplasm just splits and then changes where it goes. So it changes the probability, and it's the point is, because the way we have done this lamellipodium to grow, if it has a large surface or boundary, it will have a larger probability of growing there, right? If you have a small boundary in other places that you have a small amount of lamellipodium, you have a small amount of probability that it will grow in the other side. So if you have the same amount of lamellipodium all over, you won't break. But if there is some tiny fluctuation that makes this lamellipodium be a little bit greater here, then the probability that it will grow is larger and larger, and it takes over. And why it takes over? Because some lamellipodium grows here. It cannot grow there because of the volume constraint. Then we have the two ingredients that are necessary for spontaneous symmetry breaking, which is a local excitation, because you have the probability of growing, and a global inhibition because of the whole time. Okay? So that's the reason why it can polarize. And we made lots and lots of simulations. Some of them, they work and some they are not. The dashed line here is the same curve as we had for the experimental. So we can, I know what happens here. I know what are the parameters I have here. And then I can make by fitting I have only to discover which S, what is the value of S, and then I put there, and then I have all these natural units in terms of the simulational parameters, but I have the same thing for the experiments. So I have a translation between the simulations and the experimental units. And then I can make quantitative predictions. And these, depending on the parameters that I have used, there are some places where the simulation, the symmetry is not breaking never, and there are some other parts where the lamellipodium just detach from and they are not really. So the realistic part is like that. And This is just to show you that we can see what's happening depending on the amount of round. And then and this is I like it very much. What we did is just, okay, just to see if you are doing these things correctly, let's take something that we have not measured and let's predict what's going to happen. So we took one very detailed, one of those simulations that I presented before. It's just very detailed, and we took 2 of them here, and then we just calculated the S term, and then calculated for the simulations. All these points are the simulational points that we have obtained. This is smoothing of all the gray points, and this is the velocity autocorrelation function. Okay, so we didn't fit here. We just discovered which is the S that we should have here, and once we had the S, we just put in the same values of a scale, of length scale and time scale, and they just go one inside of the other. Okay, so we are more or less doing that. And what is nice here is that we just calculated now the modulus of the displacement, but for different taus that we are waiting. This is delta tau here as well, okay? For different delta taus for all points of many simulations here, okay? And here is the polarization, where we have defined the polarization as the difference, the distance between the center of mass of the nucleus and the center of mass of the cell. And what you see is that when a polarization is small, you don't have a very large thing. And when you have delta tau equals to 1, is persistence time. And then if you have something which is inside, not outside, in the ballistic, it's not really ballistic, but in the region that is not diffusive regime, then you can see a correlation between the displacement and polarization. But if you have delta tau, which is very small in the first diffusive regime, or very large outside the diffusive regime, you don't see the correlation anymore between the displacement of the cell and this thing. Okay? So what else have we done? Well, we have then tried to see which are the best definitions of polarization. And here, I've got some of the velocity defined at the best time. I mean, if you look here, you have all these, if you look here, you have all these correlations, different correlations with delta t, and you see when you have the best MSD, the average speeds and everything, and then you can choose the best value, the tau optimal, that you should choose to define, to measure velocity. For each different experiment, it will mean a different amount of seconds, because it will depend on the diffusion coefficient and other things of the cell. Okay? But once you have it, then you have the optimal delta tau, and then you have the polarization here and the velocity, and then you can see the correlation. That's this nice correlation between the two, which means we can define delta t, where I can see correlation between polarization and velocity, which is what we require to have in order to make the sensible measurements that we must do when we have the collective cells. So the conclusions are, if I have modified Furth equation, describes both experiments and simulations. It provides a metric to assess simulation model performance. It provides a transformation from simulation to experimental quantities. It suggests two noise sources with different time scales, probably two different biological sources. And the observation of the first diffusive regime seems to imply and internal polarization axis correlated to the cell movement direction and persistency. Okay? And what's going now? Well, we are writing the paper for Mendeley's differential equation model. Mendeley is a collaborator. And experiments to verify the hypothesis. And we have the collective motion and chemical attractants using. And just showing you, because I just wanted to use the same figure, okay? So in the first situation, we had a single cell migration in vitro, and then we have the first equation, and then now we had these measurements of MSD. Then the question is, what about the units? How to compare the results? Okay? Then we have a single cell migration in vitro, then a modified first equation, and I do the measurements, and I can make because of the modified first equation, we can now understand the measurements. But what about the differential equations? And then what we did was single cell migration in vitro and polarization, Mendeley's set of equations, measurement of MSD and polarization, which are now in course, and about how to define experimental polarization. And then that's what we are doing now, single cell migration in vitro, measuring polarization, and we are now using mounting a collective motion where we make a Stokes experiment to see the changes of velocity after the obstacle. That you see cells migrating, and then you put an obstacle, and they will have to turn around. The same thing, we have to extrapolate the Mendeley set of equations to biological fields equation, and the experiments are being of Stokes experiments. People in Lyon and in Paris are doing the experiments. We are doing the simulations. And thank you very much. Right?